Hello and welcome my dear students and biochemistry learners. Welcome to this video on definition series in biochemistry part 3. In this video, we will be studying some simple definitions about amino acids and proteins. So let's begin. Amino acids. Amino acids are the monomeric unit or the building blocks of proteins. They have a carboxyl group and an amino group bound to the same carbon atom or attached to the same carbon atom called the alpha carbon. Most of the amino acids are alpha amino acid except the amino acid proline. Essential amino acids. As the name suggests, amino acids that are not synthesized by humans and must be supplied in the diet are called essential amino acids. Non-essential amino acids. The amino acids which can be synthesized if dietary content is insufficient are termed as non-essential amino acids. Glucogenic amino acids. The amino acids that are degraded to pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate and or oxaloacetate can be converted into glucose via gluconeogenetic pathways are the glucogenic amino acids. Ketogenic amino acids. The amino acids that are degraded to acetyl-CoA are termed ketogenic amino acids because they can give rise to ketone bodies. Isoelectric pH or isoelectric point. There is a certain pH at which both the carboxyl and amino groups of amino acids are ionized, and that pH is called isoelectric pH or isoelectric point. Isoelectric point is the pH at which the molecule carries no net charge. The positive and the negative charge, they neutralize each other, they cancel each other, and hence overall charge on the molecule is 0. So there is no net charge on the molecule. Zwitterion. When an amino acid lacking ionizable R group is dissolved in water at neutral pH, it exists in solution as the dipolar ion or Zwitterion, that is the hybrid ion, which means that they have both positive and negative charges on the same amino acid, and the overall molecule is electrically neutral. Peptide bond. A peptide bond is formed by the condensation of the amino group of one amino acid with the carboxyl group of the another amino acid with a removal of a water molecule. It is also called as peptide bridge, CONH bridge. Oligopeptide. When a few amino acids are joined through peptide bonds, the structure is called oligopeptide. Polypeptide. When many amino acids are joined through peptide bonds, the product is called polypeptide. Although the term protein and polypeptides are sometimes interchangeable, molecules referred to as polypeptides generally have molecular weights below 1000 Daltons, and those called proteins have higher molecular weights. Proteins, obtained from Greek proteus, means primary. Proteins are macromolecules composed of one or more polypeptide chains, each with a characteristic sequence of amino acid linked by peptide bonds. Proteins are simply the polymers of amino acids. Moonlighting proteins. A single protein encoded by single gene doing more than one unrelated function in the cell are called moonlighting proteins. Example: aconitase acts as both an enzyme and a regulator of protein synthesis. Primary structure of proteins. Primary structure denotes the number and sequence of amino acids in the protein. In other words, the number and the order of amino acid residues in a polypeptide chain is the primary structure of protein. Secondary structure of protein. The folding of polypeptide chains into regular or ordered structure by repetitive hydrogen bonds of amino acids is called secondary structure. In other words, secondary structure denotes the configurational relationship between residues, amino acid residues, which are about 3 to 4 amino acids apart in the linear sequence. Examples are alpha helix, beta sheets and all. Tertiary structure of protein. The overall three-dimensional arrangement of all atoms in a protein is referred to as the protein's tertiary structure. Simply, tertiary structure denotes the three-dimensional structure of the whole functional protein. Quaternary structure. The arrangement of these polypeptides or polypeptide subunits in three-dimensional complexes is called the quaternary structure of protein. Example, hemoglobin has 2 alpha and 2 beta subunits. Quaternary structure is in certain polypeptides. These will aggregate to form one functional protein. So quaternary structure is assembly of various polypeptides and together they function as one protein. Disulfide bond, represented as S-S, disulfide bond. A covalent bond formed between the sulfhydryl groups of the side chain of cysteine residues in the same or different peptide chains. Denaturation of protein. The process modifying the molecular structure of a protein, that is the native conformation, without altering the primary structure, resulting in the loss of biological activity of the protein. So after denaturation, protein loses their secondary tertiary structure, lose their biological activity, and they also show increased precipitability and decreased solubility. Coagulation. When heated at isoelectric point, some proteins will denature irreversibly to produce thick floating conglomerates called coagulum, and the process is called heat coagulation. Example, coagulated egg white of boiled egg. So friends, these are some simple basic definitions in this chapter. The details about this chapter will be dealt in details in upcoming videos. So keep watching. I am thankful to all these resources for making these video possible. So thank you so much friends. Thank you for watching this video. Be happy, healthy and stay safe. And remember, biochemistry is easy. Thank you so much.